Welcome. Here is a case study that for the first time I'm sort of integrating some classwork with lab work. And my goal is to say why we do certain things the way that we do them. And a case study is a great way to illustrate the patient and then the hospital care, the doctors, and what they do. And I chose this particular one from the National Center for Case Studies because it really speaks to Chapter 16 in terms of the integumentary system, the largest uh, organ system, and also a pretty common occurrence of getting burns. Unfortunately, it's common. It's serious, a series of things that can happen. Uh, the burn alone is bad, but the treatment and identification of any infections afterwards are even more daunting. So here's the case study. Allison, she's a 22-year-old female. She was, of course, admitted to the emergency room with a burn from a campfire accident on her foot. She insisted it was not very painful and therefore waited several days before coming to the hospital. Well, there's your first mistake. That allows any infection, I don't care how big or how small, and what degree of burn, of course, the layers of the skin has been damaged. So we have a grading system, if you remember from anatomy, the various stages of burns and how serious. And if you get into where the pain receptors are actually destroyed, maybe that will speak to some of this. And then the treatments that doctors recommend. And because we don't have the direct access to the lab part, uh, a lot of times you could uh, suspect Pseudomonas aeruginosa. And one of the things that students point out is that we actually use Pseudomonas in the lab, and there's a sicky sweet smell associated with it. So, it's you don't even have to put your nose on the plate or anything. You can just smell it, and it has a greenish white color, and that actually translates into the actual bandages will have a greenish color if you have a Pseudomonas infection and those sorts of things. So there's a plethora of ways to look at things. This one did not involve, so I'll go ahead and tell you, it did not involve Pseudomonas. But let's go through the procedure of various things that happen to Allison as we go through this. Now, the doctor recommended that she receive a skin graft. Now, obviously, I chose a larger area just so you can demonstrate where you could see. It's pretty nasty, and I didn't mean to make anyone get, upset stomach-wise or whatever. But you're going to see a lot of this. So, as my dad would say, get used to it. Uh, so you can see in this first frame is pretty open. You can see this with streptococcus fasciitis and things like this. With burn patients, we see it. And then you graft in skin from some other location, or sometimes they can use artificial skin or all sorts of techniques. And in this case, it was a small area, so the graft was taken from another location, usually a leg or the buttocks area, something that won't show up in a bathing suit or something like that, and then was transplanted to her ankle. And so answer the questions that are as we go along. I went ahead and kept them in the slide set, but your slide set that doesn't have the augmentation of my voiceover gives you the step-by-step. But you can find that in the assignments area if you could respond to these, and I'll give you credit for doing this. But again, please take focus of not only the case study and as it relates to Chapter 16 and the skin and the possible infectious diseases. Even outside of bacterial, could be viral, other types of things. Look at those. But also, this goes onto the lab side. So I'm focusing sort of as a balance, but I'm taking it more to an extreme because I want to say why we are doing certain things, and so I spent a little bit more time on the lab, which you should be good with because you've done labs 8 and 9, which deals with a lot of this. And so this just dovetails so well into what we're doing. So the graft was successful, and she was sent home, and then later the graft became red and inflamed. Now, you could think about reasons why this might be so. Uh, why would there be inflammation, that sort of thing? Allison otherwise felt fine. She did not return to the hospital, which again, I would do that, especially when you've had surgery like that. It could be something systemic, some sort of septicemia or something. We got to be careful. It had spread to her entire foot. It was red, hot, and painful. So we have an immune response that seems to be localized for at least for the right now in the foot. And then she began to feel feverish and then returned back to the ER. Now, I would have been a little bit more preemptive, but this is what the case study is. And it's pretty common. I used to show slides of folks with various different types of things, and one was as simple as like jock itch or something like that. You'd be amazed what patients will live with without having to show, or drop their pants or whatever you want to say in front of a doctor, and they are willing to live with serious infections. So you're going to see this, and always try to comfort the patient, but it's just one of those things. It's really strange what a patient will do for various reasons and really put themselves at risk. And so, what would be the first line of treatment? Well, we need to know what's causing the deal. So, this is where, one of my favorite professions, of course, is clinical microbiology. And this is where we step in. And again, there's one thing I want to point out: time is of the essence. So when you're working in a clinical microbiology lab, not only do you have to know your stuff, but you have to do everything and strategize in being able to identify a microorganism to help the healthcare provider with information so that they can make choices that are effective to change the course of the disease. And we have to do that really quick, but not so quick that we make mistakes, because when you make a mistake, it could cost someone their life. So I just want to kind of bracket, as my dad would say, the boundary conditions for what we have to do here. So Allison receives her lab report, which I'm going to step through here in a minute, but that's in your packet. And if you want to, you can print that out or just view it on your screen to kind of follow along. So which bacterium do you think is responsible for this inflammation? So we have some various strains streaked on the plate, and one of which has turned yellow. So this plate is really two types. It's a differential, because we can see the differential here is mannitol is in this plate, and the mannitol is being broken down by at least one organism that's on the plate, as evidenced by the acid production and turning the pH indicator yellow, where the other two didn't produce the acid and therefore didn't break down the mannitol, and we see that as negative. So this is a positive test. These two red ones up here are negatives, and it tells us a lot, and I'm going to tell you how exactly here in a minute, but we use the differential aspect to help us identify it. So it's like a court case. Here's evidence that I'm bringing upon the preponderance of the evidence to identify the organism, and that's the way I want you to kind of think of it. And so and the other part of this plate is it's selective. In other words, it has a certain amount of salt, in this case a lot of salt, that other microorganisms just don't like, because the osmotic pressure is to the point where they can't grow. And there are organisms that can handle this level of salt that's in the plate. And with that salt concentration is 6-7%, somewhere in that range, it permits the organisms that can deal with the salt or that osmotic level where other ones can't. So that's selective. So this is a selective and a differential plate, and that's why. So here is a Staphylococcus a typical type of staph, aureus, that on a mannitol salt auger, and you can see the majority where the culture is, it's breaking down mannitol, and there's a region still sort of the reddish, that there's not enough bacteria there to change the pH. So that's what you'll see with sectoring and that sort of thing. Where the bacteria are not, it still leaves the particular color of the test media. So it's growing, so it's resistant to some degree of higher levels of salt, and it's able to break down mannitol, which helps us. Now I'm going to put this in together, but here's the diagnostic results. First, for Allison, we did the gram stain, and of course, you're familiar with the gram staining technique. That allows the the visualization of the morphology, so it stains it so we can see what the cellular level looks like, and it gives us a little information about the cell wall, the characterization of the cell wall. Gram-positive bacteria have this thick peptidoglycan and one layer of phospholipid bilayers, and that's able to absorb that crystal violet, and then is fixed into place with the mordant of the iodine, and then destain and all that, appear purple. And then the gram-negative bacteria that have very thin peptidoglycan has two layers of phospholipid bilayers, and the gram-negatives will show up, and now it takes a secondary stain to actually stain it pink. The counter stain in this case is saffron, which is typical for a gram stain. And so we wash away, because the initial stain the crystal violet did not retain, because there's no real place for it to stay, and then we counter stain, and that's what we see. Now it still counter stains with a gram positive, but the purple is so dark you don't see the counter stain. So there's the deal between the two. Now one thing I want to raise your attention to: every now and then you see some pink cells, and don't let that worry you. Oh, there's some gram negatives, this is a mixed culture. Well, actually, it's an older culture. These are dead cells, and they membranes will break down and may lose that peptidoglycan, and it won't stain with the crystal violet. And so this tells me a little bit that this was an older culture, but that's okay. You just don't see a preponderance. It's the same sort of morphology, so we're not worrying about any contaminant. Here's the mannitol salt auger, which is a differential and selective plate. And what we're looking for, of course, is a gram-positive bacteria loves to grow on it, and it does ferment mannitol, and it turns yellow as a result. So this is information that we need to process, of course, as fast as we can. And here's the final report that at certain tests, it turned the coagulase, catalase. I'm going to talk about these. The mannitol salt auger, MSA. Just to warn you, microbiologists love acronyms. You're having these 3-letter, 4-letter, 5-letter designations for everything. I don't know. I guess it's from the military influence. I'm the wrong person to ask. So the morphology, when they looked at this, gram-positive, they appeared in pairs or short chains, irregular clusters of cells, about 1 micrometer in diameter. Now, of course, they use electron microscopes and things. This is just talking about staph in general. Spherical colonies with clusters in 2 planes. In other words, the way they divide. Cell wall, very thick, peptidoglycan, non-flagellated, non-motile, non-sporing. They are capsulated, and grape-like cluster arrangement is typical. So here is an idealistic view of how these staph, and they sort of don't disconnect after they've finished dividing, and that's what gives this grape-like cluster arrangement that we've come to use. And here's a dispersal of various, you can see clustering. This was perhaps mixed a little bit too much on a slide or something, but you can see it's pretty even, and it's just kind of giving you a display. Here's a better picture, and you can see the grape-like clusters You can see the non-staining ones with the gram-positive crystal violet. These are it tends to be cells that have died and lost the peptidoglycan. So you're really just kind of staining with a counter stain the remnants of what used to be there. So anyhow, that's now the colony morphology on a nutrient auger, which you had in your kit. Nutrient auger has a golden color, and hence the name Aurium or aureus is where Staphylococcus aureus got its name, I presume. If you notice on the periodic table, gold is Au, and it's Aurium. That's the Greek name for the metal or the gold. I guess it's not a metal. It's anyhow, it gives this appearance, and it glistens a little bit, so we have that little polysaccharide that's being made. But anyhow, that's the golden color, so that's help you remember a staph anyhow, hopefully. And the other diagnostic was catalase positive. Some bacteria produce an enzyme, and this enzyme, catalase, breaks down certain products or superoxides and things like that. In this case, hydrogen peroxide. Reveals the fact that they have catalase, because catalase will break down hydrogen peroxide in a rather vigorous reaction and causes bubbling, and you'll see this. So catalase positive test is really easy to see. Now granted, you have to do this quickly. If you wait too long, pretty much if you've ever used hydrogen peroxide, eventually something will oxidize it and start bubbling. It's pretty reactive. That's why it's in a brown bottle. If you leave it on the shelf in a clear bottle, well, you'll be buying water before too long. So that's why they put it in a brown bottle. But anyhow, you can just mix some bacteria on the slide and see if it reacts. So with the case here for Allison, it is catalase positive. And we know that in a case like that, that staph is catalase positive. And this is a trick that we've known for a long time, and you can make a presumptive diagnosis really based on just this alone, catalase. Presumptive means it gives me a clue as to what to pursue, because time is of the essence. Now, I used to carry a little vial of it, or a dropper bottle, in my pocket, in my lab coat, when I was working in a clinical lab on the veterinary side. And I would just drop a little bit on a sample that I was working with, and if it bubbled right away, then I said, there's a high probability it's staph, and then I start going down that path. If it doesn't bubble, then it could be streptococcus or other things, but not staph, as far as presumptive goes. And then if it didn't bubble, I would pursue a strep, but in this case it was catalase positive. So we'll look at that. So now what we do once we have some data, and I want you to pretend you're Sherlock Holmes and you're getting data, getting ready. Of course, you'd hand it off to a district attorney or someone to try a case, but that's what you're doing, is you're trying to pin down as quickly as possible, as scientifically as possible, the identification of these bacteria. So out of the list, here's out of the case study that we had, the list, we have Staphylococcus epidermidis, Staphylococcus aureus, Escherichia coli, and Bacillus cereus. Now, I'm a big fan of Escherichia coli, but the plates are differential. The mannitol salt auger, so much salt, even E. coli won't deal with that. So we already kind of know we can rule some of these out. But I will always provide a ID chart to help you identify microorganisms. Your goal would be to understand a positive, negative test of whatever tests there are that we give you to analyze and to study. And so what you do as a detective is you start going down, well, we know it's a gram-positive, and we know it's catalase positive. So if it is catalase positive, you want to verify that it is. And then you start going down a list of things that you can look at. And one of the tests right away is that we put it on other differential type medias. Now, we already know selectively, so we're not worried about medias that are selective at this point. What we are interested in is the differentials that tell us certain characteristics. In other words, it is a characteristic of that microorganism's a biochemical trait of some kind that we can detect, or some other factor that it produces that help identify it. Now they like to stay incognito, but once they start have you start waving food under their nose, they will eat it, and then they start either breaking it down or not, and we use that information as a differential to determine. So uh, we know certain things now. Catalase, and now we know the data shows us that for Allison, her test came back with coagulase, or the organism that's causing her such a problem. It did produce coagulase. Now, it converts fibrinogen, which is the inactive version of fibrin, which fibrin is really sticky, and it starts to come together with other fibrin molecules to clot. I liken it to taking the cap off of superglue and then putting superglue out. You know that superglue hardens immediately in the contact of air. And so the fibrinogen equivalent would be what's in the tube kept from the air. But if you use coagulates and enzymes to cleaves it, it becomes active as fibrin. It's like opening the top and allowing the superglue to get exposed to the air. Fibrin now does the coagulation, and that's what you see here is this big clump of coagulation that's going on. And so it said a lot of times you add it to plasma with a bacterial colony, and you can see if the organism produces coagulase or not. And this is a big help. And the other is we can use a diagnostic tool looking at blood agar plates. And blood agar, again, is there's certain types of agar that blood you would add. It usually sheep blood or calf blood, various things, and they all kind of work the same. But sometimes some clinical microbiologists prefer a particular type of blood because it's more definitive for some organisms. But I won't go into that. So these you can see this type of homolysins that's being produced. This is of the beta type, where you can actually see through the plate and it leaves this clearing. In fact, you could see lines underneath there. I don't know where they had the plate, but you could read a newspaper through this to see it. So that's the type of clearing, or the homolysin that we see. So this would be classified as a beta. Now I've already gone over that in a previous discussion, and feel free to review that, but it's pretty straightforward. So if we go back now to our ID chart, we know it's coagulase positive. It's a typical beta hemolytic type of colony. It tells us that we're really, out of all these other things that we can do, it is a Staph aureus. Now there's certain types of tests are variations on the theme. It should give you the same result, and it does. And so we have the Staphylococcus aureus as a result here. And it's a major pathogen. It's a serious pathogen, and you're going to see it many times in your practice. So the possible pathogens have been identified in that list as Staphylococcus aureus, based on being Sherlock Holmes now, looking at the tests. This is how simple this is. If you take it, we run the test, and we run the test properly. We know how to interpret those tests, and I expect you to do those two things. But I will provide you with the ID type of information because memorizing this can be tricky, and it's not advised. I consider this similar to a standard operating procedure, where you let the data stand where it is, so it's not relegated to how good your memory is, because you have a memory a burp or something, and the next thing you know, a patient is been misdiagnosed, and bad things can happen. So we look up this data, but we have to interpret the test properly so we can follow the chart. That we need to know how to do. And a lot of the tests is very straightforward, and you can always look at the uninoculated media or tube or whatever it is to see what the non-reactive color is. In a lot of the differentials, it's a change in color. Doesn't have to be. but that's one way. And then you can see if it's a change from that. So I always insist on looking at the uninoculated as a control, just to be good. I want to rule out variables and tricks of memory and that sort of thing as best I can. So the habitat and morphology of staph of this type is consistent with what this young lady is going through. Natural habitat is mammalian body surfaces, the skin, mucosal membranes. We can see in the present in the nose and theirs, and if I were to sample you, probably I could find it. Uh, they also can be found in the pharynx. Stratified epithelial cells or mucosal types of cells, it seems to have a preference for, free sugar or cotton candy or whatever is being produced. They love it. Found in the skin, and armpits and things of humans. They act as benign or symbiotic types with hosts. Extrageneric strains of Staph aureus are found in various food products, and they can survive in dry skin. They can hunker down, and 30% of normal human healthy population is affected by staph, is asymptomatic as it colonizes. So that's pretty high percentage as a source, and that's why if you have an infection, I wouldn't wait like she did. So now we look at the biochemical characteristics of staph to really help identify it. Now, we didn't run all these tests, but if there is some reason why we can't interpret a test, or there's something, we have more tests to help nail it down. Again, it's the preponderance of the evidence. In other words, as much of these that we can throw and guarantee and convince ourselves that this is a Staph aureus. So it's catalase positive, oxidase negative. We can run a series of tests that for fermentation. Coagulase positive, as we found out. Indole negative, we can run an indole test. It doesn't produce gas. Hydrogen sulfide negative, so the media in TSI, types of triple sugar iron type of media, this would turn black, and it doesn't. Methyl red positive, we didn't run that test. Volks-Progner test is positive, and we haven't talked about it. The whole point of this is I don't want you to memorize these things, is just know that we don't rely on just a test or two. We can continue on running other tests just to confirm the notion, but a clinical microbiologist convinced, they will sign their name to the report, a lab report, and send it right on to the treating professional, the physician or whoever. In your kit you had the gelatin hydrolysis, and all that is it breaks down gelatin, liquefies it, and you can see it's positive for that. And it's beta hemolysis and blood agar. So we know this one was true. We know that the catalase was true and the coagulase was true. So those are the points. So now we shift to antibiotics. A lot of times doctors will prescribe the antibiotics. So which antibiotic do we use? There's a lot of choices out there. And the idea is that we need to deal with this pretty quickly and identify something really that's useful clinically that could help her, because it's spreading to her bloodstream. We are in serious shape. I would say critical until we get it under control. And so it's imperative that we pick the right antibiotic to use. So how do we do this? Well, there's a series of tests, the Kirby-Bauer test. There's new ones that you can use, genetic tools and the like, but it doesn't matter which of the tests you use, you want to identify the antibiotics. Now, I say that a lot of the biochemical tests or the genetic tests are more rapid. They do cost more money, but the procedure I'm going to show you can be done relatively inexpensively. It does take a little bit longer to run, but anyhow, for our purposes, it does roll down to being about the same. This is a dispenser, by the way. I'll show you another picture of that. Here's a dispenser, very much like those little pets dispensers. You rotate the head, and if you have Mr. Gorilla, whatever, I don't know why I said that, and you move the head, and the little candy comes out. Well, it's kind of like that. each of the discs. Now, all they are Whatman blotting paper cut with a sort of like a hole punch, and then soaked in a particular antibiotic, and then it's stored in these little plastic containers that are, of course, got a spring load, and you can now. And I'll show you a picture. You can release one of those discs with a aseptic uh, pair of point nose pinchers or something like that, and put them onto the auger, and you tap it just a little bit to make sure it's seated. But you can test on the surface of what? Well, you take your plate. Usually you use a particular type of media like Mueller-Hinton. The reason is when you do these standardized tests, it is a standardized test. In other words, we use the Mueller-Hinton media, and you incubate plates for a particular prescribed period of time in the incubator with a certain type of humidity, and you standardize on that, and you get these little rings or zones of inhibition, and we can actually measure those and determine if it's resistant or sensitive or intermittent in terms of its ability to be useful clinically to this antibiotic. And we make the measurement because it is standardized and it uses the Mueller-Hinton blah blah blah. We make that measurement, we can plug it into an algorithm, and it'll tell you, okay, ampicillin, let's say this was for ampicillin, and it was the best one, and it tells you, okay, we need 20 milligrams BID of this particular antibiotic, which means is you're going to give it twice a day. You're not just putting your finger in the air and just hoping it'll work. We have a systematic way now to predict what the dosing will be with an antibiotic that obviously is going to be effective, and that's the key. And this is what the patient needs. We need to know so we can treat it. Now, if we were to give this here seems to be totally resistant, so bacteria growing right up to it, that would not be a good choice. Obviously, if you were to give that antibiotic to the patient, it would be the same. Their infections would be immune to it. They just would grow, and you've done essentially nothing. And you've given them an antibiotic that's ineffective, and the patient is at serious risk. By the time you figured it out, the patient could be dead, and we don't want that. So dead is not good, just so that I make that clear. And so a highly effective antibiotic like DISC-C will produce a wide ring or zone of inhibition, and we want to verify that with using characteristic measurements. That's why you have these charts that tell you for the organism and for the antibiotic what is resistant, sensitive, and sort of in between, intermediate, so we can make that determination in our report for the patient. So we got to do this in timely, 24 hours roughly. So here's the device. If you do it manually one by one, and some of these fancy ones where it's a little clip-on actually that pops off each disc just by pushing down on that, and then you tap that to make sure it stays immediate, because we always store incubator plates upside down, and there's lots of reasons for that. One is moisture and various things, and you don't want water getting in your sample and all that. Another type is this automation type of disc. Now, what I didn't mention, and I'm going to get to that now, is we put a lawn of bacteria onto the plate. Now, there's several ways to do it. You can swab the surface. You can use a hockey stick and put a pipette, a sterile, using aseptic technique, to create a lawn of bacteria by pipetting, let's say 1 ml of a suspension of bacteria, then spreading it with a hockey stick, and then you'll get growth that's confluent. In other words, it's all over the surface, and where you put the disc, either it causes a zone of inhibition, or the bacteria just thumb their nose at you, and it's totally resistant. And so that's what we do. And the discs are always written on them as to what they are. Here's ampicillin, sulfamethazole, and various other ones. And you would take in millimeters, a measurement across the diameter, and you would report this would be 1.5 centimeters or 15 millimeters, and you plug that in with whatever the SOP calls for, and you would measure that, and obviously this is 0, and then you'd look up into the chart to see, well, is 15 millimeters uh, resistant for this particular organism and this antibiotic, or is it sensitive? And even though you see a ring, you say, oh, it's sensitive. Well, not necessarily. It's the concentration, effective dosing, and all that is taken in consideration. So here's a good candidate. You see a big wide ring, and then when you look up in the table, it in fact is sensitive to it. We would use B in this case. We could look at the others as well. You want more than one in case there's a problem. So we use Mueller Hinton Auger media, and you've got that in your kit, methicillin resistance. And that's how we know it was MRSA, because methicillin resistant Staph aureus, and this is what led them to believe originally why it was MRSA. Now, MRSA characteristically, if you have a patient that has sort of a wound that does not heal, and no matter what you put on it or whatever, it seems to persist as that lesion, then it's a high suspect that we have MRSA. And it could be an innocuous little one, or it could be a large one. Now, a burn patient, of course, is running the risk of a much bigger area, deep burn, so you're really invasive, that the bacteria sees this as like a buffet. It will start growing, and rapidly so, and produce all the things that it's going to do. But MRSA really gets in there, and it's resistant to Cefoxitin, whatever, or methicillin class of antibiotics. Now we placed them on a disc. You can see A through G or H, and you know which antibiotic it is, and then you would go through and make the measurements. And this is a pretty good sample. E and F are definitely out the door. H and B probably not, but you would run the measurements just to be sure. But C looks like a good Coloxin as a candidate. D could be, what is it? Well, C is, D is cefoxitin. I don't know. It would have to go out on the chart to see. But abscesses are one of those things, are concentrated areas. And in this case, the bacteremia grew worse. Allison's graft began to develop abscesses below it. It's very common what MRSA will do. Fills with pus. The doctor, or you have to get the Dr. Pimple Popper. No, that's just a joke. The doctor ordered tests for an antibiotic resistance, and we've now been through that. And you can see the diagnostic lab too, and that's in your kit as well. And certain characteristics of MRSA you can see. In addition to the PML, necrosis, MRSA that can cause more systemic illness. Most strains of MRSA encode enterotoxins, which cause gastroenteric and toxic shock syndromes. So this is a nasty little bug. This gets in, and so you got to deal with it pretty quickly, and that's why the pressure is on the clinician to get diagnostics running as fast as possible. We see it a lot in hospitals, prisons, military barracks, child care facilities. And again, I'm trying to give you enough of the information, 14 through 19 here, for you to answer. You're free to Google it, look it up. I do want you to, what is a PBP2A test? Well, look it up. It's a latex. I don't know what a latex test is. Well, look it up, but you'll see that it's an antibody. Anytime we agglutinate anything, in other words, a stick and clump, we're testing something very specifically. So it's not as hard as you think. Just look a lot of these things up, and I want you to get used to doing that, because technology is developing rapidly all the time, and it behooves us to constantly keep up to speed what's going on, and that requires us just to do some homework sometimes. And here's the rough part. Yeah, you're in school now, but we don't ever really leave school. So the techniques and the habits you get into now is going to follow you. So just biting the bullet, when you know obviously have to do a lot of work, you want to work effectively and learn those skills now. It's work smarter, not harder, and just come up with the tools that allow you to do these things. So the chances of you seeing a patient, well, 1.7 million healthcare associated infections occur each year in the US, resulting in 98,000 deaths, which that's pretty significant. A recent study conducted for the CDC estimated there were 94,000 invasive MRSA infections in the US in 2005. 86% were healthcare associated. That's a problem. Furthermore, a proportion of all S. aureus isolates that were resistant to methicillin have been increasing each year. So it's getting worse and out of hand, and now accounts for 60% of all S. aureus or staph isolated from intensive care units. So yes, you're going to see this, and no, it's not one that's going to go away anytime soon. So anyhow, I wanted you to be aware. MRSA is it's an issue. So I thought I'd gross you out with this. Allison's doctor first ordered complete sanitation. Usually they refer to it as abridgment, or they use some sort of way of doing a lavage of the tissue. If that's not practical, it's painful, or in burn patients, sometimes introducing other pathogens is not a good idea, and it runs the risk. So they enforce strict hand washing rules. The staff has to wear face masks, all sorts of things. They needed to be very diligent with their hand washing. Next, doctors drain the pus from the abscess. Sometimes it gets really thick and you literally pull it. It's like a string, and it comes out, and then you clean it with chlorhexidine and other types of chlorinated, and perhaps with iodine and various things, collagenase and muriprocin, Puricin, excuse me. Typical vancomycin then was applied with the wound and wrapped, and Allison was worried. Now, they use broad spectrums a lot of times when they're touching skin. We say, well, why aren't they testing? What they're doing is preventative, or they're using these preemptive type of antibiotics that are broad spectrum to stop any infections before they get started after they do a manipulation like this, because you run the risk of introducing things. And so that's what we're talking about here. And the wound was wrapped. Allison was worried about disrupting the graft, but the doctor said that IV medication was not effective on burns. Why do you think that is? Why do you think it's not effective on burns? What happens to the vascular system of different types of burns? That's why we want to learn the different types of burns, because if it's poorly vascularized, then this is the problem that we see, is that medications won't get to the site, and that's a big problem. So there's some papers to learn about. I've included two on your source there on Blackboard, learning about MRSA, and a fact sheet. Those are useful. You will see MRSA. There's no doubt. In fact, you probably already have some, either in your career professionally, or you've known someone. But there's some good references here. JAMA articles. I always like to read the emergency room type. They see things that you wouldn't believe. Anyhow, it makes for interesting, and I would keep a record of these. I wouldn't do it in paper, I would do it electronically. There are several programs out there that you can maintain a reference list that you can search. There's free ones and ones that you can pay for. Free ones are fine, and if you want a list of those, I do have some in the reference area on Blackboard. You can look up if you like. Treatment, having extreme measures, making sure that you're not going to make things worse by anytime you manipulate a wound, you run the risk And so we have to do abridgments and various things. So answer these questions. I think I've given you enough fodder, hopefully, to do that. And then healing is something you want to track. A lot of times you'll see a doc will take a Sharpie, I know, just a big old Sharpie, and they'll go around the margins of the wound. And what they want to do is to see the margins retract, in other words, moving in, and then you know we're in the process of healing. But please, these questions are really important because this will be sort of the main focus. You're the only one really that knows more about that patient than even the doctor does in terms of the progress of healing, and a lot of these reports that you're going to have to write and soap notes and the like, and then pass that information on during a nurse station change or something like that. These are the types of things that you're going to have to denote. And, is there a scab forming? Various things that are happening is evidenced by the way we see the scab forming in that, and it has a direct response as to what's going on at the molecular level. And so if you could write about that, and you can Google any of these things that you want, and that is what I have for you. I hope this was useful. We're going to do another case study that focuses on a different type of path for identification of microorganisms, a different case study, and that will pretty much conclude what we're doing. And I hope you enjoyed this time.